Hello, my name is Jose Prieto Garcia, and today we are going to talk about two hallmarks of cancer that are not that well known and that could be potential targets for future combination therapies together with chemotherapy. The first one is tumor-promoted inflammation, and the second is tumor cell migration and invasion. The learning outcomes for these sessions will be doubly. First, on the cell biology side, I'd like you to finish these lectures being able to explain how inflammation and inflammatory mediators help cancer cells to proliferate, migrate, invade, and survive. And then to be able to explain how a cancer cell acquires the ability to migrate and eventually invade all tissues. From the clinical point of view, you hopefully will be able to describe which therapeutic targets are available for advanced therapies aimed at disrupting the cancer cell migration and metastasis, two processes that are key in the natural history of the disease. Then you will be able to identify opportunities for the design and development of multi-targeted combination cancer therapies that involve maybe natural and synthetic products together. Let's start with the hallmark of tumor-promoting inflammation. Tumor-promoting inflammation, but also activating invasion and metastasis, are two of the many hallmarks that you have in cancer. Look, these are quite important from a natural history point of view of the disease. Here we have two pictures of two cells. And they are similar in that we see how these cells, rather than being with regular shapes, they appear to extend their cytoplasma in an attempt to move. This also, you can see, it's extending part of its cytoplasma in an attempt to apparently move or reach. Actually, this picture belongs to a leukocyte, which is a inflammatory cell. These are our soldiers that help us to defend ourselves from invasions, from infections, etc. But this is a cancer cell. And this is the point of this lecture, in that the cancer cell, my point is that the cancer cell actually tries to resemble an inflammatory cell. Why? Because inflammatory cells are the only cells in our body that have the ability to move across tissues. I mean, they're made in a way that they know how to walk through our body to help us where we most need it. Therefore, cancer cells will try to mimic the abilities of inflammatory cells and to use inflammation for their own benefit. So by resemble this inflammatory cell, the cancer cell will create an inflammation that will help. We will explain how an inflammation in the tumor site helps tumor cells to become cancer cells, and they will be acquiring also this ability to move across tissues, like leukocytes or polymorphonuclear cells, thus helping in invasion and metastasis. So for a tumor cell to mimic the abilities of a inflammatory cell, we need to consider how inflammatory cells are able to talk to each other. Think about inflammatory cells as being the policemen of our body. Cancer cells are the criminals, and the criminals will try to interfere in the communications of the police, let's say talking the same language and knowing the codes, so they can create a mess and they can escape through the police controls. The inflammatory cell has many ways to talk to each other. Inflammatory cells are many kinds of. We have the immune system is very diverse in types of cells, and all these cells talk a common language, which is the inflammation language. This inflammation language is based on secondary metabolites like eicosanoids, cytokines, but also and nitric oxide, but also with tools like metalloproteinases. We will see what they are in time. So these three secondary messengers are very different in nature. Nitric oxide is just an inorganic molecule with extraordinary biological capabilities. We're just starting to understand its role, and therefore I'm not going to talk much about it because it's too soon to understand how nitric oxide can be used to prevent or to favor cancer. Eicosanoids, instead, we know them very well from a lot of years of research. They are fats. They are fatty acids. And cytokines are instead peptides. Both regulate the inflammatory process and tell our body when to react or when to shut down the inflammatory with an inflammation. So we will see how these two main secondary messengers are used by tumor cells for their benefit. And as I said, we're not going to talk much about nitric oxide, which is still in some work in progress. And what are metalloproteinases? Well, these are like scissors. So, you know, to move across tissues, you have to cut them. You have to make holes. And these metalloproteinases are enzymes that degrade tissues, the collagen, the matrix, the intercellular matrix. So the cancer cell, or when it's used by the policemen like the leukocytes, will, if they acquire the ability to use this tool, they will move more freely through the body. Let's have a look at how chronic inflammation helps cancer. You know, when cancer grows, it's highly packed within a tiny place in our body. This is the tumor site. A tumor is a bunch of cells that are quickly dividing, and therefore they have an increased need on nutrients. How can they increase the flow of nutrients into the tumor side? Well, by telling the policeman of our body, the immune cells, tricking them into that there is a kind of inflammation, and when there is an inflammation, the immune cells respond with all these metalloproteinases and secondary messengers we were talking before, which in turn stimulates the blood vessels around the tumor, thus not only facilitating that the immune cells can reach the tumor, but also increasing the flow of nutrients and oxygen. So this tumor will thrive better by triggering a chronic inflammation around it, as to increase the blood angiogenesis, which is the creation of new blood vessels, and therefore all these blood vessels carrying nutrients and feeding the tumor. Well, let's see how eicosanoids can help, or well, what's the role of eicosanoids in initiating and maintaining an inflammatory process, and how this works well for tumor cells that want to become a cancer cell. Well, first of all, eicosanoids are derivatives of a arachidonic acid. Arachidonic acid is a fatty acid that is naturally stored in our membranes, cell membranes. Phospholipase A2 is an enzyme which selectively frees this arachidonic acid from the membrane. When arachidonic acid becomes available, there is a number of enzymes that will convert the arachidonic acid into eicosanoids that are pro-inflammatory mediators. The most important ones are cyclooxygenase and lipoxygenases. Cyclooxygenase is famous for converting arachidonic acid into prostaglandin E2, and this is the major eicosanoid triggering inflammation, and this is the major eicosanoid, the major target of aspirin, the most well-known anti-inflammatory drug. Cyclooxygenase also converts arachidonic acid into thromboxanes, which are used by platelets to create clots. 15-LOX converts arachidonic acid into 15-HETE, so does 12-LOX into 12-HETE, and 5-LOX will convert arachidonic acid into leukotrienes, which are most well known. Let's say that that leukotrienes and prostaglandins are the two major pro-inflammatory mediators. The rest are less important maybe in our story. All these mediators not only regulate inflammation, but have been demonstrated to have roles within the story, the natural evolution of cancer. For example, prostaglandin E2 will favor that a primary tumor becomes an inflammatory site, and therefore this inflammation will bring more nutrients and new blood vessels to feed the tumor and create and favor the proliferation of cancer cells. Thromboxanes may help the invading, the freely moving tumor cells into disguising to successfully circulate around in our bloodstream until they reach any organs where they can colonize and create another site. However, for example, 15-HETE seems to be a good guy that will block this thromboxane action. 12-LOX in turn looks like helping the cells that got into our bloodstream with the help of thromboxanes into extravasating, leaving the blood to proliferate into an organ. And leukotrienes seems to favor, for example, in the case of melanoma, the metastasis into new organs. It seems that Cancer cells have more chances to establish themselves in other organs such as lungs with the help of leukotrienes. In the lab, we could experimentally block the activity of cyclooxygenase with normal non-anti-inflammatory non-steroidal anti-inflammatory drugs like aspirin and meloxicam, or with experimental molecules with this that inhibit 15-LOX, a natural product that is present in many plants which inhibits 12-LOX, and some experimental drugs that inhibit in different ways 5-lipoxygenase. In the lab we can test all these inhibitors and check whether these reduce or increase cytotoxicity, whether these affect migration of the cells, invasion, and finally metastasis. These are really the views we have in the lab when we work with cancer cells in all these experimental models of cytotoxicity, migration, invasion, and metastasis. You will agree with me that that this is a very complex view. I don't want you to know perfectly all of these and all these interplays. I need you to focus on that we have cyclooxygenases and lipoxygenases, that they may have different roles in cancer migration and invasion and tumor promoting inflammation. And now we are going to just focus on COX and 5-LOX, which are much more important. Let's start with cyclooxygenase and prostaglandins, which are probably the most well-studied aspects of tumor promoting inflammation. As I said, arachidonic acid may be metabolized by cyclooxygenase, but we have two isoforms, COX-1 and COX-2. COX-1 is naturally always expressed in our cells. COX-2 in turn is always expressed when we have inflammation or cancer. COX-1 is necessary for many homeostatic functions, such as keeping our stomach without ulcers, protected from acids, and that's why when you take aspirin, one of the common effects is that you block the activity of COX-1 and you get ulcers because you are inhibiting a protecting activity of COX-1. We would like to have NSAIDs that only target COX-2, and these are the like of meloxicam and other CAMs. But in turn, they have shown some sort of cardiac toxicity. Well, so when we look at this picture, then we understand that we would like to target COX-2 rather than COX-1. COX-2, as I said, is overexpressed in practically every pre-malignant and malignant condition. It has been proven to be highly expressed in colon, liver, pancreas, breast, lung, bladder, skin, stomach, head, neck, and esophagus cancers. And this overexpression of COX-2 has been clearly associated with promoting angiogenesis, creating new blood vessels within the tumor, so feeding the tumor as a collateral, and also helping invasion. It is true that it is also related with apoptosis, and we may target COX-2 also to directly kill the cells in a programmed manner. As I said, the other important eicosanoids are the leukotrienes that come from the metabolism of arachidonic acid by the 5-lipoxygenase. It is a little bit more complex in its pathway, where the arachidonic acid go to the 5-lipoxygenase, which acts in conjunction with other enzymes, other proteins, and produces several intermediate leukotrienes that go out and act on receptors. And therefore, we would like to see how whether we inhibit the synthesis or we inhibit the receptors. Would it be better or worse towards designing cancer therapies? Well, we will have a look. As I said, this enzyme, lipoxygenase, is the key enzyme for all this pathway. It creates what we call leukotrienes, but they are attached to some oligopeptides, and they're called cysteine leukotrienes. The overexpression of 5-LOX has been described very specifically as a vital feature of prostate cancer cells. For example, there is a cancer cell line which is PC3, and when you inhibit the enzyme, the cell dies by apoptosis. So we are here looking at a potential application of targeting the eicosanoid pathway to help in the therapy of certain cancers. However, the COX-2, as we said, it's virtually overexpressing almost any malignant cancer. So 5-LOX is much more specific in targeting certain cancers. COX-2 may be a kind of cure-all cancers, but unfortunately it's not that the case, as we will see. Let's have an overall picture of the tumorigenic effects of both prostaglandin E2 and leukotriene B2. These are very complex, again, mechanisms, and I only show them with the intention of you looking at the differences rather than I don't want you to learn all these pathways. I don't even wouldn't know how to reconstruct it from my head. But the important thing is that look, when we have a tumor and they start releasing prostaglandins and leukotrienes, as I said, when they start learning the language of inflammation, they trick all our immune cells into creating a tumor supporting microenvironment, right? As a result, we have more blood vessels going into the tumor, and we facilitate the proliferation, the survival, the migration and invasion, the angiogenesis and immunosuppression, thus helping cancer protection. That's the overall effect of these two messengers. But there's a difference between the two. When prostaglandin E2 talks to the receptors in a cancer cell, what it does is, after a number of mechanisms, is activating the proliferation of the cancer cell, is helping the cell to migrate and invade, and is enhancing its survival chances. However, leukotriene B2 when interacting with its own receptor in cancer cells, apparently has a more restricted effect. The overall effect is restricted to proliferation and cell survival. So the difference here is this, that prostaglandins help all features, but differentially from leukotriene B2, are key to cell migration and invasion. And this is extremely important, because the difference between a tumor and a cancer is that cancer cells move. Cancer cells are tumor cells that have learned to invade. And then that's the difference. When a tumor cell starts to move and invade, then we are in front of a cancer. So then again, the overall picture of these two messengers is quite different, with prostaglandins and prostaglandin E2 in particular being able to promote all features of cancer cells, whereas leukotrienes are more restricted and always help in proliferation and help survival, but not into the invasion and migration that characterizes cancer. So this is a more tumorigenic effect, and this is a more oncogenic effect. Okay, so what has been shown in clinical trials using COX and LOX inhibitors? Did they successfully do anything in tumor promoting inflammation? Well, let's have a look to them. We have had first clinical trials looking at inhibiting COX-2, second clinical trials inhibiting both COX-2 and 5-LOX, and third clinical trials inhibiting only 5-LOX. When we were inhibiting COX-2 only, it looks like patients, for example, with advanced melanoma who were treated with COX-2 inhibitors have a significantly lower rates of melanoma outcome measures. So it looks like, yes, inhibition of COX-2 is a potential area where we could add NSAIDs, as I said, non-steroidal anti-inflammatory drugs of but COX-2 selective together with a chemotherapy like Taxol or other chemotherapy drugs. Together with them may help in improving the outcome of cancer patients. When we were trying both, inhibiting both pathways in certain cancers, we didn't see much of an improved outcome. For example, this study in non-small cell lung cancer failed to demonstrate the value of this inhibition. However, when they went back to the results, they looked at patients who responded well to this experimental therapy, and they realized that these patients were those with a higher COX-2 expression. So again, we may see that COX-2 expression is a much important, not only a biomarker of malignancy, but also a biomarker of potential successful treatment with NSAIDs added to the chemotherapy. As I said before, 5-LOX is more restricted into being highly expressed in cancers. It's more specific of prostate tumors, for example, and yes, there were some studies trying to see whether the proliferation of these cancer cells was much better or much lower when introducing COX inhibitors, and also they tried 12 inhibitors, but unfortunately they were 12-LOX inhibitors were ineffective, whether 5-LOX were effective. So we see that, for example, cancers with high expression of 5-LOX, like prostate, could be maybe successfully treated when adding an inhibitor of 5-LOX to the chemotherapy. So let's now talk about cytokines or chemokines, like sometimes are defined or referred to. My message to take home with chemokines is that they are very multifunctional mediators, and they're incredibly they're incredibly general in their effects. They impact all the tumor processes, but also they act in inflammation in a way that is very, very complicated to rationalize, and we're only starting to understand, unravel all the interplays they have. As a result of these pleiotropic effects, the only thing we can say is that the overall outcome of the tumor development depends on a balance. Imagine this is a balance in between pro-inflammatory cytokines and anti-inflammatory cytokines, because we have both. There are cytokines that are triggering inflammation and cytokines that are inhibiting inflammation. Depending on where the balance is tipped to, for example, having a few chemokines of this type, we will have limited inflammation, then restricted vascularization, and therefore tumor growth will be more or less restricted. However, if the balance goes to a lot of pro-inflammatory chemokines, like this here, there will be a greater inflammation in the tumor site, leading to more creation of new vessels, new blood vessels, and a more rapid tumor growth. So we will be favoring. Obviously, if we alter the balance toward more excessive inflammation and angiostasis, let's say to an extreme that the inflammation is extremely high, we will have a surprising effect, that this extreme important inflammation brings in all the immune system cells to kill the cancer cells, and they will kill the cancer cells. So what you want is a balance, not too low, not too high inflammation, just the right inflammation to get food, but not to call the attention of the immune cells. And this is the entire trick we are talking about in the hallmark of tumor promoting inflammation. The cancer would like just to call in the police, but not too much as to get caught. Just the balance, the right balance to get a continuous efflux of food, but not an entire army coming and clamping on it, like it would happen in this case. So the thing is that chemokines as a target, and this is the message to take home, is promising, but it's quite complicated. And I don't want you to learn all these details. What I would like is to express that yes, we could pinpoint several chemokines that could be cancer for certain carcinomas. But the problem is that despite so many, there's quite a lot of experimental clinical trials on it, and we're still trying to rationalize which chemokines can we use to target different cancers. There's no still a clear therapy using chemokines, but we are trying, and the message to take home is that there is an active research on it, that obviously chemokines are the agonists and antagonists are much more difficult to create than aspirin and all the other antagonists or inhibitors of eicosanoids. They are much difficult to produce, and each company can invent a different one that can be patentable. So to summarize all this, we are looking at a balance. We need a balance in which we inhibit the immune system, but we don't immune depress the patient, as well as the tumor wants a balance in between how much inflammation wants to trigger, not too much, not too low as to not get enough food, enough to get food, but not too much inflammation as to call upon them the entire army of our immune system. So the Eicosanoids on their own so far failed to bring inconsistent results. However, when we combine them with other chemotherapy drugs in patients that are overexpressing, for example, COX or 5-LOX, then we just have a good outcome, and this is something we want to pursue in the future. That with chemokines, all current studies indicate promising roles. The problem is that there are a number of antagonist inhibitors that are still experimentally tested, and we don't have still a clear way to go with this part of the immune system messengers. Right, so let's talk now about tumor cell migration and invasion. As I said, migration and invasion are the two key features that make a tumor to become a cancer. A cancer cell is a walking dead, is, let's say, a tumor cell that can walk. There are different types of ways to walk, and so cancer cells try to mimic the way leukocytes walk or move across tissues in a more or less perfect way. We have different types of migration for the tumor cells, which are first the perfect one. This is the perfect way to walk. This is when the cells learn to move via a 5-step migration cycle that is exactly the same way immune cells move across the body. Remember that to acquire all 5-step migration cycles by just random mutations, where you all know that cancer acquires hallmarks by more or less random mutations, it's quite a difficult case. It's very rare that a cancer cell really acquires the mesenchymal migration feature, but when it does, it's a highly dangerous cancer that knows how to move perfectly across your body. Then we have a less perfect way to walk, the amoeboid. This is when they move like amoebas or like a blob of jelly. They more or less slip through the tissues. They don't know how to walk well. It's like a baby crawling. So in the amoeboid migration type, it's a less adhesive type of and less elegant type of expansion and contractions that in the end lead to some movement, but it's far from perfect and it's far from coordinated. Nevertheless, it may contribute to tumor cells to invade. And these are the ways one tumor cell is moving, but they can move as groups also. These are chain migration and collective migration are when the cells are moving as a pack. And if it is one cell that learns how to move and drags the others, then we are in front of the chain migration. There are chain, and the leading cell is the one which has acquired maybe a quite perfect way to move and drags the others. Whereas the collective migration is led by a group of cells that more or less know how to move, and they drag the rest of the cells behind them. Let's try to understand how this perfect movement, as I said, the mesenchymal way to move works. As I said, there are 5 steps or 5 cycles in this way, or 4 cycles, depending how you see it. This is an overview of how things work. You see, there's a phase where the cell attaches to the tissue and then extends itself. So that's the 1, that's the 2, that could be the 4 cycles. Then the cell extends, then the cells adheres the extension, then the cells destroy the tissue so they can penetrate across, and then they move. Let's say I would have to make the new cell here, and they move, they escape from the tissue, and then they invade other ones. For the cell to extend, a cell that attaches to the frontier between tissues, this is tissue 1 and this is tissue 2, and this is the ECM, the extracellular matrix. This is the frontier between tissues. When the cell adheres or attaches to the matrix, then it's going to extend itself to move forward, and then not only we have to adhere by focal adhesion, but we have to stretch the cell. Stretching the cells mean actin polymerization. Actin, as the name the name implies, is an active. Actin for active protein. It's a protein of the movement, provides some traction force within the cell to stretch the cell in the direction of the movement. Actin polymerization is regulated by the RAC proteins, which in turn belongs to a GTPase family. I'm not very keen in you memorizing all this, but I would like you to memorize rather that cell extension relies heavily in actin polymerization, and that by targeting those pathways to which actin polymerization belongs, then you could target the cell in this first step and stop it. Let's see the second step, the adhesion. When the cell anchors, stretches in the form of movement, now we need to anchor the part which we stretch it out, and this is by means of what we call focal adhesions. To anchor the cell, the second part, the stretch part of the cell to the matrix, we need an anchor, and anchors have chains, and chains are catenins. Catenins are proteins. Catenin in Latin means chain, catena. Catenins are essential proteins in anchoring the cells. So it's very important that the cells anchor well, because this is a very slippery matrix. The matrix is collagen and cartilage that are very, very slippery proteins. And the cells need to anchor well. It's like you have walking through a slippery floor. You could easily lose loss adhesion and fall, so the cell needs to really adhere and in order to be able to move. If you are in a slippery in a floor full with oil, you would probably slip, fall, and you would be unable to really move because this oil would would prevent you to anchor, to have a hold of the ground. So I'd like you to remember this, that the stretch part of the cell needs to anchor as well as it did here, and this is done with chains that in turn are proteins called catenins. Right, let's have a closer look at this. These molecules, these catenins, are depending on some adhesion molecules also, which are called cadherins. And we have two kinds of cadherins, the E cadherin and the N cadherin. And the E cadherin means epithelium. And the epithelium we know is, for example, a skin is an epithelium, and the cells in our skin are well attached, right? This is because they have a fully functional E cadherin. Cancer cells don't want to, they want to attach, but not to attach too much, because if they attach too much and become glued to the tissue, they cannot move further. So they need catenins to anchor transiently to the external medium, but they don't want to have E cadherins that they would attach forever the cell to it. So they mutate the E type to the N type, which is a non-functional cadherin, and so they have changed to attach, but they don't have cadherins to attach forever. So that's why they can't move, because they are deprived. This mutation took them from having E cadherins to N cadherins. Right, once they are attached to the matrix, they want to destroy it to cross. How do we do this? Well, now we are talking about these tools, as I said, metalloproteinases, which are like cutting tools. They are proteinases, so they split proteins. They hydrolyze all these proteins, this collagen, this which is in the extracellular medium, and then once they open up a hole, forget about this. Once they open up a hole, they can squeeze through it and invade the next tissue 2. All right. Once, as I said, they made a hole, they have to contract, to squeeze through it. How they contract is by developing a contractile force, which depends on some fiber assembly and contraction promoted by the rock. I always remember this protein because the movement of cancer cells is like a rocking chair. You actually rock forward and backwards, and by doing it with some force, you advance. It's like if you have a rocking chair and you rock hard, maybe this rocking chair is owing not to stay in the place but moving across the place. So that's why I remember this. RHO associated kinases are helping the malignant cells to squeeze through these holes by contracting the actin filaments they developed before. And at the very end, as I said, they want to detach, squeeze, contract, and then go out of this hole and detach from the border of the tissues to become freely moving in the tissue number 2. This detachment is due to a concerted action of a number of proteins. It's quite complicated process. To mention a couple of them, there's the FAK and the ERK pathways are playing a role in this, but well, I'm pretty happy for you to remember that after the contraction you need a detachment from the extracellular matrix that is a concerted action among many proteins. So if we put together all the molecules that help movement in one map like this one, it becomes pretty much complicated, but also means that if you want to target the movement of cells, you may have many Achilles heels. You may have many molecules that come, for example, this molecule is targeting here and here, so they may multi-target different proteins that help the movement. So we are in front of a process that could have potentially so many targets, and many of those targets could be multi-targeted at the same time by the same molecules. This is what we are pretending nowadays, to look in deep at the different targets, the different controlling proteins of the movement of cells to stop in their tracks. The limitation of targeting the movement of cells is that cancer cells will, if you handcuff, let's put this image for you, if you handcuff the cancer cell from moving, let's say for example you chain the right leg of the cancer cell eventually will try to find a way to use the left leg to move, and so on. If you allow me this image, once you inhibit one process within the cancer cell that leads to movement, they will try other processes to allow for maybe a less elegant movement, but a movement anyway. This is what we call plasticity, the ability of cells to avoid barriers by developing other skills. So that's the limitation of anti-migratory agents. We may multi-target many, that's why it's important to multi-target as many proteins that participate in the movement as possible, not only one. As I said, one of the metalloproteinases are somehow key, because if you don't make holes in the border lines in between tissues, the cells cannot invade another tissue. And there's a lot of research going on this field, trying to inhibit different metalloproteinases that are have an enhanced expression in several cancers, like for example metalloproteinases 9 is very much expressed in non-small cell lung cancer. There have been the development of really fancy things like DNA enzymes that can be engineered to block the complementary sequence of the target genes in metalloproteinases, but nevertheless we don't have yet any breakthrough with metalloproteinases. There's an enormous amount of research going on in this aspect, but so far no success with one of these molecules having any preclinical promise. Maybe you, some of the people that is listening to me, maybe they will get into this field and eventually discover something. I hope so. As I said, for at least 30 years there has been promising targets. So far, no preclinical, all the preclinical studies say these are lead compounds. Clinical trials have been disappointing. So I'm afraid this is the end of these 2 sessions that I just stuck together for convenience for you. We've been looking at targeting the many steps of cancer natural history, from the primary tumor to the proliferation, the invasion, the circulation, the strabisation, and the metastases are potentially being modulated by both eicosanoids and chemokines. And that this is a highly complex process where chemokines is about a balance of getting the, targeting. The tumor cell wants to target the right balance of chemokines as to get not too low, not too high inflammation to thrive. And also we have to look at which chemokines favor this and to inhibit them. And also that there is a division in between prostaglandins and leukotrienes, in that prostaglandins favor all hallmarks of cancer, including the migration and invasion, which is the difference between tumors and cancers. But leukotrienes are much more restricted to, let's say, prostate cancers and only proliferation and cell survival. We've been looking at how the process of movement acquisition happens in tumor cells to become cancer cells. Remember, tumor cells don't move, cancer cells move. And we were going through the many steps of the perfect way to move for a cell, but we then also concluded that maybe you need to target as many of these processes, because if you only target one, the cell will try to learn how to move without this being possible, which is called plasticity of the cancer cell. The cancer cell is very plastic. It learns how to evade our therapies. So I hope you got both a new and an interesting view to the 2 different aspects of cancer, which is tumor inflammation, tumor promoting inflammation, and movement. These are aspects that are less known. Probably the aspects you know more is how to kill the cell itself, but not how to stop the inflammation that benefits the cells or how to stop the movement. If we are going to exploit these 2 processes, we always have to add the killing drug. So this would stop the cell, the cancer cell, but would not kill the cell, or only in very few cases you would kill the cell. For these 2 hallmarks of the cancer to be successful in clinical trials, they have to be administered together with the killing drugs, without the chemotherapy. We don't like them, but we don't have other option right now. So thank you very much for your attention, and any doubts, please reach me via email. As I said, what I want you is to not to learn all these mechanisms and pathways. Just the most important names are eicosanoids, chemokines, metalloproteinases, know what they are, know the difference between prostaglandins and leukotrienes, and keep in mind that blocking this tumor inflammation promoting tumor promoting inflammation and movement of the cells will not solve the cancer. These therapies would have to be combined with chemotherapy as to kill the cancer. By then,